My name is Khateriaa Pyrtel, and I'm a genetic counselor at the Siteman Cancer Center. Welcome to our video series. Today I'm going to speak with you about BRCA1 and 2 deletion duplication testing. The specific instructions for each of our genes is contained within our DNA, which can be thought of as sort of genetic building blocks. Our DNA is represented by the letters A, T, C, and G. Through the work of many scientists, we have a pretty good idea of what letters of DNA we should expect to see in each of our genes. Genetic testing is a method of medical test which allows us to identify any potentially harmful changes in our DNA sequence that would impact the way a gene functions, and these types of changes are considered mutations. Two frequently used methods of detecting genetic mutations are DNA sequencing and DNA deletion duplication analysis. As more people undergo genetic testing and we learn more about the possible mutations that can exist in a gene, we've discovered that both DNA sequencing and DNA deletion duplication analysis may be required to pick up the largest possible number of genetic mutations. One specific genetic test which we have discovered both methods are required are in genetic testing for the BRCA1 and BRCA2 genes. The vast majority of mutations in BRCA1 and 2 are small genetic changes involving only a few bases of DNA and are readily detected through the DNA sequencing method. However, over time, we found that in as many as 10% of families who have a mutation in BRCA1 or 2, these mutations are large, involving several DNA bases, and are detected through deletion duplication analysis, are in fact missed by the sequencing method. Although these larger mutations account for a small percentage of BRCA1 and 2 mutations overall, we do know that in some ethnic groups they are in fact more common than in other ethnic groups. If someone has had BRCA1 and 2 genetic testing recently, it is very likely that their testing included both the DNA sequencing and the DNA deletion duplication analysis method as well. If someone is uncertain if their testing included both methods of analysis, it is recommended they reach out to the medical provider that ordered the testing or to the lab that performed the testing to see what methods of analysis their testing included. If the DNA deletion duplication method was not included, this part of the testing is available as a standalone test and costs much less money than the DNA sequencing portion of the test. Because these larger genetic mutations can also increase someone's risk for breast and ovarian cancer, it is recommended that for those individuals who had sequencing only and were found to be a mutation, that they consider having this testing as well, because this type of mutation would also warrant changes in medical care, including increased screening and possibly preventive surgery in these families as well. Thank you.